Hello, I'm Christine Curcio. Thank you for listening to this tutorial. This is the second of 4 tutorials about the extracellular deposits of AMD. It is helpful to hear the overview tutorial first. This one is about Drusen-driven AMD. Here are my supports and disclosures. My takeaways for the 4 tutorials are these. AMD is a solvable 4-dimensional puzzle, visible in vivo via OCT-anchored multimodal imaging. The photoreceptors and RPE together are a sensory chip of the brain, including the geometry of the foveal singularity. We discussed the value of a 3-layer versus a 5-layer Brooks membrane, and that the essential AMD pathology is extracellular deposits on either side of the RPE. Drusen followed the cone vision system, and subretinal drusenoid deposit, or SDD, begins where the rods are numerous. One hypothesis incorporating the deposit lipid composition is that they both represent failed outward transport of lipids, mostly those taken up from diet, to maintain the membranes of cones and rods. My takeaway for this tutorial is that drusen are a late life manifestation of the cone visual support system, and they confer huge risk for AMD progression. High-risk drusen are direct precursors to type 1 macular neovascularization and atrophy. They can be differentiated from low-risk panretinal drusen. Their localization under the macula lutea at the fovea within it is an important key to mechanisms. Risk from aging is huge and equal to all gene variants and lifestyle together. Drusen occur from relatively functional RPE and impaired transport of constitutively released materials, including lipoproteins. The lipoproteins from RPE lead to inflammation and a cellular and humoral response. Cell culture model systems exist, and therapeutic approaches from cardiometabolic disease can be explored. The works of Sarks, and Killingsworth were seminal in studying dozens of eyes of clinically characterized and longitudinally followed AMD patients. Transmission electron microscopy, like this, was a primary technique. In their clearly described and annotated horizontally oriented panoramic electron micrographs, it was possible to see that OCT is coming. Good electron microscopy, such as this view of well-preserved contents of high-risk soft drusen, are useful for current and future interpretations of clinical OCT. In this light micrograph, we see the druse as a lump and its continuity with a thin layer of the same material called basal linear deposit, or B-Lin-D, and they're both visible in clinical OCT. To review, the OCT-B scan on the left shows layers affected by AMD. The schematic shows a progression from normal aging on the left to advanced AMD on the right. The reflective bands arise due to the precise horizontal alignment of millions of vertically compartmentalized cells. There are multiple biologically distinct layers within 100 micrometers vertical height between the ELM, the dashed red line on the top, and the choriocapillaris endothelium on the bottom. These include the inner segments of the photoreceptors, the outer segments, the interdigitation zone between the outer segments and apical processes of RPE, the RPE cell bodies, Brooks membrane, and choriocapillaris together. These bands may be visible, perturbed, or even absent in an AMD eye. Further, layers are added representing the extracellular deposits. This is SDD between photoreceptors and RPE, B-Lam-D between RPE and its native basal lamina, and drusen between RPE basal lamina and the inner collagenous layer of Brooks. In this section, we will see how drusen are the direct precursors of advanced AMD. The Sarks showed in 1997 how soft drusen material was found in the same tissue compartment as choroidal neovascularization. This is schematized in this cartoon. In the histology, we can get a close-up view of soft drusen here with a little bit of artifactual separation. B-Lin-D is in the same sub-RPE-BL space. A few hundred micrometers away is a breach in Brooks through which a neovascular complex with activated pericytes pass through, leaking fibrin, we believe. Between these two locations, there is sub-RPE basolaminar fluid that is continuous with both the drusen and the exudate from the neovascular membrane. The availability of OCT made it possible to see in the clinic what Shirley Sarks had described years ago. That is, neovascular complexes under the RPE that did not leak fluid. This is called non-exudative MNV. Here's a B scan on top showing an RPE elevation. Histology showed a type 1 MNV that had stayed stable with 20/30 vision for 11 years. The overlying outer retinal bands representing the photoreceptors are intact. As mentioned, Drusen and B-Lin-D are pro-inflammatory. This immunohistochemistry shows nicely signal for complement factor H in both the age normal Brooks membrane on the top and in high-risk drusen in the central area on the bottom. It's possible to see that it is the central area by the shape of the fovea and the thick Henle fiber layer. Immune cells were first found in the sub-RPE basolaminar space, indicating a cellular response by Penfold and the Sarks in the 1980s. Here, cells are shown in drusen for an eye with neovascular AMD. You can see the giant cells and the macrophage is filled with lipid droplets that are distinct from the overlying RPE. An important missing piece of the AMD puzzle was how drusen led to atrophy, and this became clearer with OCT. Let's look at this large drusenoid pigment epithelial detachment, or PED. In the B scans, the RPE starts out as smooth and even. At 7 months, RPE cells can be seen to leave the top of the PED and migrate inwardly. By 23 months, the streaks of hypertransmission show the loss of shadowing by RPE. Then the drus collapses and atrophy ensues at 41 months. This life cycle can be quantified easily in the B scans. Drusenoid PED grows slowly, then collapse quickly. Smaller soft drusen have been shown to do this too. We interpret this data based on what I will show in the next few slides as follows, that drusenoid PED grow because RPE constitutively produces material that normally exits to the circulation, but is impaired from transit by aged Brooks and choriocapillaris. The PED collapse because production by RPE is discontinued and the clearing processes, whatever they are, can catch up. So then, by color fundus photography, atrophy begins because drusen collapse, but now, by OCT, drusen collapse because the atrophy begins. Another way to look at this is that the RPE is relatively healthy, doing its jobs, even on top of large drusen, which form because transport across Brooks and choriocapillaris is impaired. Recently, clinical imaging has shown that drusen height is strongly related to features of atrophy, which fits with this model. Let's see how one small drus leads to atrophy. In the upper right are B scans showing disappearance of a drus and development of a hypertransmission streak. Below are 2 histologic sections through this area. At a higher magnification of an off-axis cut, the ELM skims over the top with the green arrow, and 30 micrometers inferior is a slice through the apex of this drus. The RPE and photoreceptors are both absent. On either side of the apex, the green arrowheads show how the ELM descends. An ELM ring slides down the slope of each individual drus. The arrow shows Mueller cell processes sweeping downward. Over time, or perhaps even at this time in a different section, we expect that these processes punch into the drus, as we saw in other drusen. So our working hypothesis is that atrophy begins atop individual drusen. Why are some drusen considered high risk? We learned this through longitudinal studies of large populations. The Beaver Dam Eye study, led by the inestimable Ron Klein, reported the frequency of early AMD lesions in over 4,500 people. They used color fundus photography and the ETDRS grid, which you will remember from the first tutorial. In this table, they showed the age-adjusted frequency of early AMD pigmentary changes and drusen stratified by the ETDRS subfield. When area is accounted for, the small central subfield, all cones, is disproportionately affected. In fact, the ratio of early AMD in the central subfield to the outer ring was an amazing 26. It drops by a factor of 6 from the central subfield to the inner ring. The authors offered no explanation of this foveal concentration, and they did not talk about it in their subsequent work. The population-based Blue Mountain Eye study from Australia, using similar methods, supported the original Beaver Dam observation with longitudinal data. The odds ratio for progression to late AMD at 10 years due to drusen at baseline within the central subfield of the ETDRS grid is again 26. Aging is much bigger than the other population-level risk factors. In fact, all the risk gene variants and lifestyle together gets an odds ratio of progression in about the same range. For comparison, smoking, an agreed upon risk factor, the odds ratio is about 3. The low risk in the outer ring of the ETDRS separates the central soft drusen from the many drusen in the eye. In other words, it captures the margins of this high-risk area. The ARIDS2 group recently confirmed that there was no added risk from outside the ETDRS. And finally, there is a threefold drop-off within the inner ring, so the risk is highly concentrated within the central 3 millimeter diameter macula lutea. This slide comes from our recent study combining data from 2-wavelength autofluorescence for macular xanthophyll pigments with histology and longitudinal epidemiology studies. This image on the left is typical of 30 normal-aged eyes. The xanthophylls are highly concentrated in the central subfield of the ETDRS grid, extending into the inner ring. We use the grid to compare maps of soft drusen locations measured in OCT and the topography of AMD progression risk from the Blue Mountain Eye study that I just showed you, and the topography of potential mechanisms in the neurosensory retina. We see that the concentration of drusen is high and that the progression risk is related to the concentration of the xanthophyll pigment. The cones are highly concentrated in the center, but the profile of progression risk is broader than the cone peak, as is the abundance of soft drusen. These are better matched to the macular xanthophyll pigment. What about histology? The Sarks-Killingsworth group said that high-risk soft drusen containing membranous debris were found only in the central area. Here's an example of membranous debris from our own material. Our microdissection study directly compared central and peripheral drusen, 415 total, and indeed we found soft drusen only centrally. Other studies considered seminal in their time could not make this determination because they either sampled the peripheral retina or combined the central and the peripheral retina. We differentiated among all the small RPE elevations in our online repository. The top row shows nodular drusen of different sizes. These are panretinal and they take up fluorescein, as shown by other studies. The bottom row shows soft drusen in panel D and basal mounds in panels E and F. This is soft drusen material trapped inside B lambda. This material raises the RPE, but is expected to have a different risk profile from the sub-RPE basal lambda drusen. This slide from the overview tutorial compares findings in 20 and 90-year-old normal central retinas along 7 tissue-level measures. In the middle is a 2-fold thickening of Bruch's membrane and a 15-fold increase in the esterified cholesterol, the main ingredient of lipoproteins. Let's focus on that huge increase. In this next section, we will consider what Drusen features confer high risk for progression in tissue-level studies. This schematic provides a roadmap for the next section. Lipoproteins accumulate especially in the intercollagenous layer starting in late adolescence. Then they accumulate between the RPE basal lamina and the ICL as pre-basal linear deposit, which was originally called lipid wall. Lipoprotein particles fuse into lipid pools in basal linear deposit, and they pile up in drusen. We called this the oil spill on Bruch's membrane, or more accurately, the oil spill on Bruch's membrane. And it's most easily explained if we consider aging changes in Bruch's and choriocapillaris as initiating events. The net effect is a barrier to transport and a source of pro-inflammatory lipids. It's only partially visible clinically, a point I'll come back to later. To characterize and quantify extracellular deposits, we created a website of AMD histopathology called Project Macula. 132 short postmortem eyes collected over 14 years were processed for high-resolution histology and electron microscopy preparation viewed by light microscopy. Sections through the fovea and perifovea were systematically reviewed and photo-documented. Thicknesses of chorioretinal layers and the contents of the subretinal sub-RPL spaces were measured and annotated at pre-defined locations, as shown below. The histology, sampling scheme, and photomicroscopy were designed under the hypotheses that the photoreceptors drove the metabolic demands on the vascular support system and that the signal sources for OCT are subcellular. Soft drusen and basal linear deposit, B-Lin-D, are 2 forms of the same high-risk material. Soft drusen is a name given from color fundus photography. These represent a lump and a layer, and pre-basal linear deposit is an even thinner precursor. Together, these form a transport barrier that puts the RPE into a state of metabolic insufficiency. Drusen lipids can be peroxidized to create pro-inflammatory intermediates, and by virtue of biomechanical fragility, they form a cleavage plane for invading choriocapillaries. As drusen grow, they move the RPE away from the choriocapillaris, adding to RPE hypoxia and stimulating migration into the retina, like we just saw. This slide shows the continuum of sub-RPE basolaminar lipid built up over adulthood. Here's a soft druse, basal linear deposit, pre-basal linear deposit, which has a non-undulating profile, and no sub-RPE basal laminar lipid because the age 3 layer Bruch's has lipoproteins. In 2 eyes of 1 clinically characterized donor, only 20% of the lipid in this critical compartment were drusen, the clinically visible part. This comparison of many AMD and age control eyes comes from the Project Macula website and shows that soft druse material is more common in the fovea than in the perifovea, and that the precursor pre-basal linear deposit is also more common in the fovea than the perifovea. So drusen appearance in AMD culminates a process starting in late adolescence and ongoing throughout adulthood. This slide summarizes how we studied Bruch's membrane lipoproteins that constitute a major component of soft drusen. We hypothesized that the Sark's term membranous debris did not capture a formative mechanism for the abundant nonpolar lipid that accumulates with age. We use Philippine histochemistry to label cholesterol forms. Esterified cholesterol is released by cells only as the correlative lipid of lipoprotein particles, and this localizes exclusively to Brooks membrane. Unesterified cholesterol is found in the surrounding cells and interesting at lower concentration in the outer segments. We use conventional transmission electron microscopy on top and quick freeze-deep etch on the bottom to show pre-B-Lin-D as a layer of particles with surface and core morphology, and B-Lin-D on the right, in which particles are fused to form lipid pools. Let's also remember that the RPE expresses hallmark genes of a lipoprotein secretor. This slide shows the same pattern of Philippin staining for esterified and unesterified cholesterol in Drusen of AMD eyes. And in the same eyes, we saw immunofluorescence for apolipoprotein B in the drus on the bottom and basal-linear deposit on top. ApoB is the principal backbone protein of a large lipoprotein. In this section, we address the question of whether diet is the main driver. We will posit that the lipids in drusen could result from uptake from plasma or outer segment phagocytosis or endogenous synthesis. And we're going to follow the lipids in clinical, compositional, and cell culture systems. This case report shows dramatically that outer segments, or even the whole retina, are not required for drusen formation. Over 7 years, this patient with a macular hole, that gap in the retina, also had drusen forming and reforming in the gap. So it is possible that the RPE is taking up material from plasma, even without signaling or from the retina. For the compositional studies, let's look at these fatty acids, which are long hydrocarbon chains that attach to a terminal carboxyl group. Fatty acids are components of phospholipids in all cell membranes and all esterified lipids in lipoprotein particles. They're also precursors to signaling molecules with wide-ranging bioactivities, including inflammation. Important polyunsaturated fatty acids in the next few slides are 18, 2, and 6, or linoleate, which is the most abundant in the body, including plasma, and 22, 6, and 3, docosahexaenoate, or DHA, the most abundant in retina. We can thus probe fatty acids for clues as to their origin. We isolated Brooks membrane lipoproteins and compared them to plasma lipoproteins. Esterified cholesterol had abundant 18,2 linoleate and very little 22 and 6 or DHA. The phospholipids were also rich in 18,2 and low in 22 and 6. The high ratio of linoleate relative to DHA in Brooks membrane lipids was also shown by other labs. This ratio resembles plasma. Thus, we concluded that diet is the main source of fatty acids in Brooks membrane lipids. In the next section, we will talk about how the lipids in Brooks membrane can become pro-inflammatory. In this part of a molecular circuit diagram, we see drusen and basal-linear deposit in numeral 3 and a nidus of inflammation at 4. In 1999, Rick Spade and Don Armstrong showed that isolated human Brooks membrane has chromatography signals for peroxidized fatty acids, the principal being peroxidized versions of linoleic acid at the arrows, 13S-H-POD and 9S-HOD. These compounds were prominent in other inflammatory conditions, which is why they looked. In 2 studies, first in rabbit, then in rat, by the Luddy lab, subretinal injection of these toxic lipids caused choroidal neovascularization. This was the first means to produce CNV experimentally beyond VEGF injection and mechanical disruption of Bruch's membrane by laser. It's also possible to detect these molecules with antibody E06, which is specific to phospholipids containing oxidized fatty acids. These were detected in Bruch's membrane of both human eyes and on mouse eyes on the bottom. Another form of peroxidized lipid, plausibly from lipoproteins, is 7-keto-cholesterol, which can form by both enzymatic and non-enzymatic means. Ignacio Rodriguez isolated 7-keto-cholesterol from carefully collected monkey eyes of all ages and found much higher content in RPE choroid than in other eye tissues, and this went up with age. It was also high in isolated drusen. Ignacio's lab also provided the first in vivo evidence that this molecule was pro-angiogenic in a corneal neovascularization model using rat eyes. It's possible to see inflammatory cells in the sub-RPE basolaminar compartment where drusen are found in human eyes. In this eye with neovascular AMD, we see macrophages with lipid droplets resembling foam cells from systemic arteries. We also see giant cells, which are fused macrophages. This makes sense as macrophages are constantly surveying this region from the vantage point of the choroid, as shown by the Luddy lab. An important component of high-risk drusen appear to be lipoprotein particles. In the next few slides, we will review some of the essentials of this biologic system. It is fortunate that we can learn a lot from what has been already done in liver and intestine. In this schematic of systemic lipoprotein metabolism, we see a route for exogenous lipids from diet. These are taken up by the intestine and repackaged as chylomicrons to deliver fats to the tissues and to the liver. The endogenous lipoprotein system starts with VLDL at the liver, which delivers fats to the tissues, and we are very familiar with LDL, which is a remnant particle from VLDL created by enzymatic remodeling in the plasma. So in the systemic system, the liver is the coordinating center, and the RPE has characteristics of both the liver and the intestine. So it performs these functions in the eye. Lipoprotein particles like that in the upper left are multimolecular assemblies that solubilize oil droplets for transport through an aqueous environment. The core is rich in nonpolar lipids, and the surface has phospholipid, unesterified cholesterol, and apolipoproteins. Plasma lipoproteins like LDL and HDL are the best characterized. However, lipoproteins are also released by the brain, the heart, placenta, kidney, and we also think the RPE. Lipoproteins are specified genomically by their apolipoproteins, such as ApoB, ApoA1, ApoE, and so on, alphabetically to ApoO. The atherogenic particle number is the number of ApoB containing particles of all sources in plasma. This is now considered a strong predictor of risk for the disease of coronary arteries in the heart. Of the lipoprotein components, cholesterol is either unesterified or free or esterified to fatty acids. There are many ways to release unesterified cholesterol from healthy cells. There is only one way to release esterified cholesterol from a healthy cell, and that is in the core of a lipoprotein particle. We were able to isolate from Bruch's membrane of humans a cholesterol-rich ApoB and E containing lipoproteins of intraocular origin with inflammatory potential. The esterified cholesterol source were these large particles that were solid, spherical, 80 nanometers in diameter with a surface and core structure. The same fraction contained ApoB and these other proteins. The size and flotation properties resembled hepatic VLDL, but the lipid composition, because it was so EC rich, resembled LDL a remnant particle of VLDL. The RPE has capacity for lipoprotein assembly and release. Microsomal triglyceride transfer protein, or MTP, is required for lipoprotein assembly and release. Here is signal for both ApoB and MTP in human RPE in a location consistent with endoplasmic reticulum. Secretion of full-length ApoB has been achieved in this rat-derived RPE cell line. We showed particles in culture, and they were EC-rich. Similar findings were also reported by Jim Handa for the human RPE19 cell line. Thus, our scenario is that pro-inflammatory lipids that could promote AMD progression are in Bruch's membrane, and these originate largely from lipoproteins released constitutively by the RPE. This pathway has been studied in detail for hepatic VLDL. A major difference is that the RPE lipoproteins are EC-rich and not TG-rich like VLDL. Much progress has been made in animal models of this pathway from laboratories of Irina Piccoliva, Kathy Bose-Battaglia, Jim Hurley, Jason Miller, and others. The idea that the RPE is a constitutive and polarized secretor of ApoB and E lipoproteins is the basis of a response to retention hypothesis of AMD based on a concept of the same name proposed for atherosclerotic cardiovascular disease by Ira Tabus and Kevin John Williams. We called it the oil spill strategies because of the lipid and because most people would agree that cleaning up an oil spill is a good thing to do. We suggested three approaches: skimmers and dispersants to scavenge oil spill lipids, a top kill, which is to modulate the outflow of RPE lipoproteins, and a bottom kill, which is to modify the dietary intake that provides so much of the lipid to these lipoproteins. There has been progress regarding the sequestration of oxidized phospholipids or cholesterol in Bruch's membrane. An apolipoprotein A1 mimetic peptide has been shown effective in removing histochemically detectable lipid and restoring Bruch's membrane ultrastructure in mouse, and additionally reducing membrane attack complex, the terminal effector of the complement pathway, in monkey eyes. Regarding the lipidation of ApoB-containing lipoproteins, several approaches are being investigated, including local delivery of statins, reduction of ER stress, and increasing fatty acid oxidation. Regarding the dietary input to RPE, the ARIDS2 supplement of lutein and zeaxanthin can be viewed in this context, as well as low glycemic diet, Mediterranean diet, and probiotics. My UAB collaborators and I are pursuing this hypothesis that lutein and zeaxanthin supplements may directly impact drusen formation in this manner, that the RPE takes up lutein and zeaxanthin and other important essentials on LDL and HDL from plasma at receptors on the basal surface. The RPE transfers L and Z to the retina for use by photoreceptors and Mueller glia. Then the unneeded lipids are repackaged and released in lipoproteins of the RPE origin, and drusen form as choriocapillaris and Bruch's age blocking exit, and the risk for type 1 macular neovascularization increases. So if plasma HDL and LDL are enriched with lutein and zeaxanthin from supplements like the ARIDS2 supplement, it's possible that the RPE takes up fewer plasma lipoproteins, it releases fewer lipoproteins of its own synthesis, drusen are smaller as clearing mechanisms catch up, and risk for neovascularization decreases. And this is what was shown in the ARIDS2 trial. So I hope that I have convinced you that drusen are a late life manifestation of the cone vision support system that confer a huge risk for progression. High-risk drusen are direct precursors to type 1 macular neovascularization and atrophy. They can be differentiated from low-risk or panretinal drusen, and the localization under the macula lutea, or the fovea, is key. The risk from aging in the form of drusen is equal to the risk conferred by all the variants of known genetic associations, plus lifestyle together. We can think about this as functional RPE and impaired transport together make drusen. Lipoproteins from the RPE could be a source of inflammation to which cells in the neighborhood respond. Cell culture model systems exist, and therapeutic approaches from cardiometabolic disease can be explored for AMD. Here are the key takeaways from this 4-part tutorial on deposit-driven AMD. If we can prevent atrophy from starting, we will also prevent its expansion. And if we can prevent neovascularization, we can also prevent fluid and fibrosis. Deposit-driven AMD is one way to think about these goals. Thank you for your attention.